If you can just, Ryan, if you can just stop sharing.
Yeah, I'm trying to figure out how to do that. Stop sharing. Okay, I got it.
Wonderful. Lena, can you try to share your screen? We can't hear you. Please open your microphone.
Oh, okay. Is okay?
Yes, but now we would like to see a presentation.
Okay. Can I start?
I don't see your presentation. I don't know if anybody else does. Good. Good. Okay. Yeah.
Okay. Now I will my start. Okay. Thanks. Okay, thank you. Hello everyone. It is my great honor to share our research work in this conference. Today, my research topic is about that self-fusion between MSC and lung cancer cells contributes to the formation of lung cancer stem cells. First, let's briefly understand the background of self-fusion and cancer. As we know, self-fusion is a process that two or more cells become the one cell by fusing their membrane and nucleus. Self-fusion is also one of the common phenomena in cancer development and progression. Studies have shown that cancer cells can fuse among themselves or with other different types of normal cells, such as macrophages, stem cells, and stem cells, and so on. More and more studies have shown that self-fusion contributes to cancer progression, including aneuploidy formation, tumor initiation, acquisition of drug resistance, increased proliferation, and higher metastatic patient. So self-fusion is also considered as a new mechanism of cancer metastasis. MSC is a multipotent stem cell that has the capacity to self-renew and differentiate into diverse lineages, such as bone, adipose tissue, and muscle. MSC is also a critical component in the tumor microenvironment and also a putative fusion candidate. Now, MSC is often used in self-fusion study. As we know, MSC has four typical features, such as strong cell-like properties, stem cell-like properties, high metastatic ability, and tumorigenicity. Many studies have reported that MSC can involve in cancer progression through self-fusion. However, the underlying molecular mechanism of MSC fusion with cancer cells remain poorly understood. For example, several studies have shown that stem cell enhances molecular characteristics upon fusion with cancer cells, but some conflicting reports revealed that stem cell reduce tumorigenicity through cell fusion. So the effects of MSC fusion on cancer growth and metastasis are still controversial. It is well accepted that EMT is an essential step of cancer metastasis. The occurrence of EMT is accompanied by the expression changes of many EMT-related markers. For example, the epithelial marker E-cadre is downregulated, while the mesenchymal markers, including uncadre, vimentin, and fibronectin, are upregulated, as well as many EMT transcription factors, such as steel slug twist and DBY2. In addition, many signal pathways have been proved to be involved in the regulation of EMT. It mainly includes TGF-beta, 1-beta-catenin, notch, NF-kappa B, ERK, and AKT signal pathways. Cancer stem cell is a subset of a cancer cell population within tumors that possesses self-renewability and participates in cancer initial progression, metastasis, and recurrence. However, the origin of cancer stem cell is still ambiguous. A recent work published in Cell Journal has revealed that EMT can generate cells with properties of stem cells. Many signal pathways involved in EMT regulation are closely related to the self-renewal of cancer stem cells. At present, a new hypothesis of the origin of cancer stem cell is cell fusion. Differentiate cell fusion with stem cell can generate the cancer stem cell. However, the molecular mechanism of the cancer stem cell formation and cell fusion is not very clear. So, in our study, we will investigate the function, roles, and molecular mechanism of cell fusion between MSC and cancer cells. So we established the cell fusion model between MSC and lung cancer cells to address the molecular mechanism of cell fusion in lung cancer growth and metastasis. On the other hand, our research objective was to verify whether the hybrids between MSC and lung cancer cells are lung cancer stem cells. So to solve the above this problem, we first established cell fusion model between MSC and lung cancer cells LRC in vitro using PG induction and flow cytometry sorting. In brief, MSC was touched by GFP expressing vector, while the LRC cells were touched by RFP expressing vector. To generate the fusion hybrids, cell fusion was induced by PG in mixed LRC RFP and MSC GFP stable cells. After cultivation for 24 and 48 hours, the hybrids were identified by fluorescent microscope. Next, we identified the biological characteristics of the hybrid cells. Karyotype analysis showed that the average number of chromosomes in hybrid cells was significantly more than LRC cells. To determine whether the DNA polyD was changed after cell fusion, so we analyzed the DNA contents of the hybrid cells and parent lung cancer cells by flow cytometry. The results showed that LRSC were typically diploid, while the fused cells were aneuploidy. Meanwhile, the cell cycle progression of the hybrid cells was arrested at the G0 and GY phase. We also found that the P21, P27, and P53 were increased in the fused cells compared with lung cancer cells. So these observations suggested that cell fusion may inhibit cell cycle progression by retarding the phase transition from GY to S. Next, to test whether the growth ability of lung cancer cells was influenced by fusion with MSC, we first performed CCK8 experiments, and we found that the growth rate of hybrid cells was markedly lower than LRC cells. Meanwhile, the growth ability of hybrid cells was also determined by plate colony formation assay. Consistently, a severe reduction in growth rate was observed in fused cells. To evaluate the tumorigenic potential of the hybrid cells, we also performed the soft agar cloning formation assay. The results showed that the hybrid cells grew slower and formed fewer clones in soft agar. So these results indicated that MSC fusion can induce growth inhibition and reduce the tumorigenic potential of lung cancer cells. To test whether the metastatic ability of hybrid cells was changed in vitro, the word healing assay and transwell migration and in vivo assays were carried out. The results revealed that the hybrid cells had greater migration and in vivo abilities than that of in the parent lung cancer cells in vitro. Next, to investigate whether cell fusion between MSC and lung cancer cells affect tumorigenicity in vivo, we established xenograft tumor model in nude mice. Tumor formation was observed for about 4 weeks. As shown in figure, the tumor volume and tumor weight of the hybrid cells showed no significant changes compared with LSC cells. Meanwhile, we performed another experiment to determine whether cell fusion affects cancer metastasis in vivo. Cancer metastasis was evaluated by pulmonary nodule counting, weighing, and cytopathological staining analysis. We found that the number of pulmonary nodules in the fused cell group was significantly increased in comparison with that of in the LRC cell group. Cytopathological staining analysis further confirmed that the hybrids showed stronger metastatic capacity. Taken together, these results suggested that cell fusion between MSC and lung cancer cells enhanced the lung cancer metastasis in vivo. Okay. Morphological observation showed that parameter lung cancer cells and MSC cells display rounded and elongated morphologicals, respectively, typical of epicellular and mesenchymal cells. After fusion with MSC, the hybrid cells lost epicellular morphology and exhibited a fibroblast-like appearance with elongated shape. These changes reflected that the hybrid cells may undergo an EMT because EMT play an important role in generating cancer stem cells. So, to validate whether the hybrid cells acquire cancer stem cell-like properties, we detected the stemness of the hybrid cells. As we know, CD133 and CD44 are the surface markers of lung cancer stem cells. Our results showed that the MIR levels of CD133 and CD44 were markedly increased in the fused cells. Furthermore, we observed that the expression of many stem cell transcription factors, such as OCT4, anoxos2, TF4BF1, were significantly increased in the fused cells. In addition, we also observed that the multi-drug resistance genes of tumor cells ABCG2 and ABCC1 were obviously upregulated in the fused cells. So this data provide evidence that the hybrids derived from MSC and lung cancer cells acquire stem cell-like properties. Our findings further indicate that Stem cell fusion induces the formation of cancer stem cells. Due to the morphological changes and the enhanced metastatic abilities of the hybrid cells, we hypothesized that the hybrid cells might undergo an EMT. To explore whether cell fusion results in an EMT, we examined the expression changes of many EMT-related markers by article PCR and Western blotting assay. We found that the epicellular marker was downregulated, while the mesenchymal markers, including uncatarated alpha SMA fibronectin, were upregulated. Meanwhile, many EMT transcription factors, including COS, LAG, AB, and FOX2, were also obviously increased in the fused cells. So, also, this data provides strong evidence that the fusion hybrids between MSC and lung cancer cells acquire stem cell-like properties by undergoing EMT. At last, we investigated the expression of MLP2 and MLP9. Our results showed that the hybrid cells exhibited an obvious increase in the expression of MLP2 and MLP9. So taken together, our observations indicate that fusion of MSC promotes lung cancer cell migration and in vivo, while the induction of EMT and upregulation of MLP2 and MLP9. On the other hand, our data also demonstrates that fusion of MSC with lung cancer cells can acquire the stem cell-like properties and generate lung cancer stem cells by undergoing EMT. In conclusions, our findings provide strong evidence that fusion of MSC with cancer cells can generate cancer stem cells by undergoing EMT. Cell fusion hypothesis provides a direct link between EMT and cancer stem cells. This study also provides a new explanation for the origin of cancer stem cells. Okay. Many thanks to our group members of BJOT and our collaborators of Jilin University. This research work was supported by the National Natural Science Foundation of China. Okay, that's all. Thank you. Thank you for your attention.
Thank you very much, Nina. There is one question here at the chat from Farah Hussein. Thank you for your nice talk. What about the genetic instability and chemoresistance of fused cells compared to diploid CSCs? Can you see this? Can you see the question?
Okay. Can you speak wise? Thank you.
Can you see the question on the chat?
Oh, I don't see.
So I will read it again. The question is whether, what about the genetic instability and chemoresistance of fused cells compared to diploid CSCs?
Yes. We found that LRC were typical diploid, but while the fused cells were not typical diploid, but the DNA poly was in disorder, but not the genetic information will be changed after fusion with MSC.
Okay. Thank you. Other questions?
Yeah, may I ask a question?
Yes.
I noticed you use PG to induce the cell fusion in tissue culture. So is any evidence that this fusion also occurs in cancer tissue in vivo? And if it is, what can regulate those stromal cells, cancer cells fusion in vivo?
Okay. In fact, some literature have reported that they observed the cell fusion in vivo. There are some literature has proved the cell fusion phenomenon can occur in the tumor tissues. But in our study, we just do the experiments in vitro study, not in tumor tissues to observe the cell fusion phenomenon. Because we choose to PG induction is that the cell fusion occurrence is very low frequency. If we don't add the PG induction, it is about less than 1%.
Okay, thank you.
Thank you very much.